Thank you. Well, I'd like to start by thanking the organizers for inviting me and giving me a chance to share some of our recent work on the Dog Aging Project, and also just the opportunity to be in a room full of experts in veterinary medicine and people who are especially interested in veterinary gerontology, which is not a common thing to find. So I'm really delighted to be here. I also want to thank in particular my co-author on the proceedings paper that we wrote and my collaborator for the past decade on the Dog Aging Project, Kate Creevy. I just spoke with Kate earlier. She said that she knows a lot of people here. Those of you who are in the room, you know who you are. Kate says hello. So I'm going to start with a little bit of background, just some very general background about aging and how I got involved in it and how it led me to the study of aging in dogs. And then I'm going to tell you 4 stories. The first 3 are about the kinds of work that we've been doing recently, measuring aging-related disease in dogs, looking at determinants of healthy aging, some studies that we're doing to actually intervene in aging. And then finally, I'll tell you about the Dog Aging Project, which is still very much in the planning stages, although we have some preliminary studies that are underway, and I'm excited to share our plans for the future with you. So throughout my career, which started about 30 years ago in aging, there are 2 overarching questions I've been interested in. First of all, why is it that some species live longer than others? And we have a range of species, and some of these are longer lived than you might think. For example, these eusocial ant species, which might be just a centimeter long, the queens can live for up to 30 years. The longest lived animal on this slide is actually not the whale, although some of you might have said that. It's this little clam, Arctica icelandica, which can live at least for 400 years. The other big question that I'm interested in is why is it that there's variation within populations? Why is it that some people age faster than others? To ask that question, the first thing we need to think about is how do we measure aging? We certainly can see it all around us. We see these physical changes in ourselves, in our loved ones. There are many different ways of measuring it. This example I'm giving you here is sort of an interesting example. It's an extreme measure. What I'm showing you is the world record for speed of running a marathon. In blue are males, in red are females as a function of age. And there are a few things that you see here that are kind of interesting. The first is that aging with respect to the world record fastest speed of the marathon happens actually, it doesn't start until our late 30s. Many other ways we start aging in our early to mid 20s. After age 20 or so, males are always faster than females. There are some 90-year-olds that are capable of completing a marathon, and there are some parents crazy enough to let their 6-year-olds run marathons. That doesn't capture the variation, though, that I'm interested in. And here's another marathon data set. These are data from a cross-country ski marathon, and looking at speed as a function of age. And again, you can see a steady decline with age, but then there's also a lot of variation above and below that line. That's what I'm really interested in. And that variation comes from 2 sources, and this is something that I'll come back to throughout the talk. 2 and only 2 sources, genetics and environment. Genetics captures the potential to be able to complete a ski marathon, and to do so quickly, that you inherited from your parents. And then the environment captures all sorts of things. What you ate for dinner last night, the training that you had done over the past few years, and whether you were lucky enough to be born in Sweden or Norway instead of somewhere like the US. The Swedes and Norwegians are probably up here. Aging is kind of the great connector if we think about disease. All the major killers in human adults have as their single greatest risk factor, in some cases by many orders of magnitude, aging, not lack of exercise or cigarette smoking or diet. Age is the single greatest risk factor for many, many different diseases. And this is a fact that really motivates this huge area of research in the biology of aging, or what we now call geroscience. The thinking is that if we can somehow understand the factors that moderate aging and actually slow it, we can potentially affect all these different causes of aging and so increase health span. And that's the goal. The ultimate goal is not to make people or dogs or other organisms that live forever. The goal is to make sure that our later years are as healthy as possible, to increase the amount of healthy years. We've certainly seen dramatic increases in health span over the past 200 years. So what we're looking at here is life expectancy from 1840 to the present. And you can see this dramatic increase over time in many different countries. Most of this impact is due to our ability to deal with early life causes of mortality, especially infectious disease. But of course, we've also seen great success in the past 100 years in our ability to deal with causes of late age mortality like cancer and cardiovascular disease. So this is great. We've seen enormous impacts in human aging, but understanding how that happens and what we can do for the future is really difficult. The problem is that humans live for just too long to really get at the mechanism. And this is partly what motivates my own interest in thinking about dogs. So I want to tell you a little bit about how I got here. So I started out as a comparative biologist in graduate school, studying lifespan across different species. And as you can see here, the single greatest predictor of lifespan across species is size. The relationship between longevity and size is strongly positive, but there are other interesting things going on. So for example, these blue points are birds and mammals that are flying. Flying species tend to be longer lived than terrestrial species. And there are other ecological factors as well. This, of course, isn't new. It's something that Aristotle pointed out in 400 BCE, and Francis Bacon in 1600 pointed out that big species live longer than smaller species. So we've known that for a while. This is what I worked on early in my career, trying to understand why, and then I got into fruit flies. Fruit flies are a terrific model. On the left, we've got a mortality curve for humans. So we're looking at the log of mortality as a function of age, the risk of dying at any age, for males in blue on top and females below. And then these are three different fly populations with mortality curves that look very much like human mortality curves if you just substitute day for year. So flies are really a great model. People have been studying aging in flies going back to the 1920s. We've learned a lot. One of the things, though, that's missing from flies and worms and yeast and mice, which are the four major species that we focus on in studies of aging in the lab, is variation. So this picture here is a famous photograph taken at Connecticut State Agricultural College about 100 years ago. These are recruits, or students, the cadets rather, lined up from left to right by order of height. And you see this nice normal distribution. This photograph is famous among quantitative geneticists because it illustrates that there is variation, that it's frequently normally distributed, and that to really understand variation in a population for any trait, we need to understand the sources of that variation. And thinking about aging, which is an incredibly complex trait, this is what got me excited about dogs. And in particular, I realized that dogs just might be the answer. When this paper came out, now more than 10 years ago, in 2007, from Elaine Ostrander's group, and I saw the cover of this issue of Science showed up in my mailbox. I hadn't even opened it up, and already I thought, I think we need to start working on aging in dogs. This particular study was an attempt to understand the genetic basis of variation in dogs. And of course, we've got a Chihuahua and a Great Dane side by side. Dogs are the phenotypically most variable species on the planet. Not only in terms of size, but we'll start with size. And Ostrander, who's now at NIH, realized that this gave us an opportunity to dissect the genetic basis of size. And one of the really interesting things about dog size and why I was so excited to see the cover of this issue of Science is that dogs are interesting when it comes to lifespan. As those of you who work with dogs know, the relationship between lifespan and size is opposite to what we would expect. Large dogs tend to be short-lived compared to small dogs. So remember, across species, little voles and mice, shrews are down here. Large species like elephants and whales are up here. Dogs show the opposite relationship. In fact, the number one gene that predicts size in dogs is IGF-1, or insulin growth factor 1. What we're looking at here is the frequency of the big A allele in this study in Science as a function of size. And you can see that for most of the small breeds, they're actually fixed for this one particular allele of IGF-1. Most of the large breeds are fixed for what is the ancestral allele, the same allele that we find in wolves from which dogs descended. So there's this very nice relationship between the frequency of IGF-1 and size. This is really interesting from the perspective of aging, because for years, people have been interested in the relationship between IGF-1 and lifespan in many different species. This is a paper from worms, mice, humans. So there's great interest in IGF-1 and aging. Of course, I saw the results in this paper and I thought, gee, I wonder if IGF-1 is the reason why large dogs are short-lived and small dogs are long-lived. We don't yet know the answer, although people like Kimberly Greer are working on that question. But it really motivated my interest in starting to think more deeply about aging in dogs. And in particular, to try and solve this particular equation. This is a fundamental equation. Don't worry, it's the only equation I'm going to show you in this talk, and it's a pretty simple one, although it's deceptively simple. P stands for phenotypic variation. So these values here are variances, distribution of traits. So here, the P is any phenotype that you could possibly measure. It could be height, it could be lifespan, it could be blood chemistry. G is the genetic variation. E, of course, is the environmental variation. And then G by E illustrates the fact that certain genotypes can have an effect on phenotypes that depend on the environment. So a particular genotype might make some animals live longer than others, but only in the presence of a particular diet. So it's complicated. And of course, within genetics, there's lots of complication: additive effects, dominance effects, epistatic interactions between genes, and so forth. So dogs are really the perfect species in which to solve this mathematical equation. The payoff is not just that we understand some fundamental things about the nature of aging, but of course that we can help dogs. And what better payoff? So, of course, dogs show tremendous variation. I have a whole lot of slides that I took out of this talk because I don't need to convince this audience about all the different kinds of variation that dogs show, including morphological variation. But it's important to remember that there's also tremendous environmental variation. It's hard to see. This is a dog right here in front of its doghouse. And of course, so there are rich dogs, there are poor dogs. These dogs don't have a doghouse. There are healthy dogs and not so healthy dogs. That environmental variation gives us a wonderful opportunity to understand how environmental variation shapes healthy aging. And we have sequenced genomes. In fact, one of my many collaborators on the dog aging project is Eleanor Carlson, who's at the University of Massachusetts Medical School and the Broad Institute in Boston, and she now has 700 fully sequenced genomes that we're working with. So lots of genetic information. And of course, people are already using that information to map traits like cancer risk in golden retrievers. So the groundwork has been laid for the kinds of work that we want to do. We need the phenotypes, though. We need the data. And what we've started, and what I'm going to tell you about for the next 2 stories, is some of the phenotypic information that we've been collecting using existing data sets. So I'll start by talking about just our attempts to understand variation, not in terms of morphology or behavior, but aging and age-related disease in dogs. I'll talk about morbidity and frailty. So as soon as I saw this paper, I thought, wow, I want to work on dogs. I'd had some earlier conversations with a colleague, Steve Ostad, whose wife is a veterinarian, and she and Steve had told me about the veterinary medical database. So I remembered that. I called up Steve. He said, yes, I think I can get it for you, but I didn't know anything about veterinary medicine. So I called up the University of Georgia Vet School, where I was at the time, and after 3 or 4 phone calls, I finally realized the person I needed to speak with was an internal medicine specialist, Kate Creevy, and that was one of the best connections that I've made with my career. We've ended up continuing to work on this project for about 10 years. So the first thing that Kate and I did with one of her fellows was go through 80,000 records of deaths in the veterinary medical database that occurred over a 20-year period. And for each record, we had the breed, we had the age, we had its sex, and we had all the diagnoses. In some cases, the diagnoses were uninformative with respect to cause of death. We just knew that we just had information that the dog was euthanized. But in some cases, we had a large number of diagnoses from which we were able to define the actual cause of death. And our first paper was really just a description of the spectrum of age-related diseases that occur across a large number of breeds. For the common breeds, this was information that veterinarians already had, but we had large numbers of dogs, large numbers of individuals for even rare breeds that one wouldn't see regularly in a practice, like Affenpinchers, for example. So the kinds of things that we could see, and this is just to illustrate visually the scope of the variation for 4 different causes of mortality: respiratory, cardiovascular, hematological, and gastrointestinal. So of course this bulldog, the mascot when I was at UGA, the most common cause of death was respiratory. For this Chihuahua, it's cardiovascular. For a retriever, hematological, and then for this Shar-Pei, gastrointestinal. And of course, this is just a little slice of the tremendous amount of variation that we saw and continue to see as we continue to work with these data. We were also able to look at comorbidity. This is something that the National Institutes on Aging is very interested in. And I need to point out to this audience that historically, as those of you who are in veterinary science know, and as I discovered as I began to meet veterinary researchers, NIH has not been a great funder of veterinary research. This is changing, certainly with the comparative oncology group at NCI, but also NIH is now really excited about funding aging research in dogs. And that's something that I'd be very happy to talk with you during this meeting. If you're interested in, I could give you the names of the program officers at NIA who are excited to talk to people who want to work on aging in dogs. One of the things that the folks at NIA want to figure out is the relationship between aging and comorbidity. We know that the number of chronic conditions increases as we age. This figure here shows life expectancy as a function of the number of chronic conditions. And you can see that for 67-year-olds, those with 10 or more chronic conditions have a life expectancy of 4 or 5 years. Those with no chronic conditions can expect to live at least to 90 years old. So the number of chronic conditions is highly associated with life expectancy. What's not clear is which way the causal arrow goes. This is something that NIA wants to figure out, and we think that the dog is a terrific model to figure that out. So for example, here are 2 analyses that we did of dogs from the veterinary medical database and also dogs from VetCompass, a data set that Dan O'Neill has collected through veterinary practitioners in the UK, and this is data held at the Royal Vet College in London. And in both cases, what you can see is that there's a large distribution of number of diagnoses So this is the number of diagnoses plotted on a log scale. It's a straight line, which means it's exponentially distributed. Most dogs at death just had 1 or 2 or 3 diagnoses, but there were some who had up to 30 or more. Part of the variation is intrinsic. Part of it, in this case, had to do, remember, all these dogs were at a vet school. Not all of them were necropsied. Those who were necropsied might have been necropsied by a very well-trained senior pathologist or by a new vet student, so the number of diagnoses might have been very different accordingly, although we see similar patterns in the vet compass data, which is dogs not necessarily at death, but just number of diagnoses of dogs coming into vet clinics. The underlying forces that are shaping these distributions, this one being Poisson, which is similar to the exponential if plotted on a log scale. Here it's a linear scale. We don't know what those causes are, but it's certainly something that we're interested in working on. Importantly for this audience, there's obviously going to be a nutritional component. This is something that I'm very excited about working on, one of the reasons I was so thrilled to be invited to this meeting. Here are, from the vet compass data, comorbidity by diagnosis. So the average for the entire population is 3 chronic conditions. And it's notable that among dogs with obesity, it's more than twice that. I've had the experience of flying on airplanes and sitting next to people who ask what I do and telling them about this work, and they ask about nutrition, and sometimes the topic of diabetes comes up, and they say, well, of course, diabetes isn't a problem in dogs, and I just love to feed my dog, and I know better now to keep my mouth shut. Obviously, it is a problem, something we need to be thinking about. I'm really excited about working on dogs, as dogs, for dogs and dog owners and veterinarians, but people are obviously excited about dog as a model to inform our understanding of human aging. There are some ways in which dogs and humans are really alike. If we look at the proportion of people at any age dying of a particular cause, here we're looking at cancer. So the proportion of individuals that die of cancer in humans and in dogs with age, and you can see a similar pattern, an increase and then a late age decrease. This is due to ingestion of toxic substances, poison or drugs. And again, it goes up early in life and then comes down in both humans and dogs. And then there are some striking differences. For example, cardiovascular disease in humans, #1 killer late in life, but of course in dogs, very few die of cardiovascular disease. And interestingly, there's very little effect of age. Some people have suggested that this makes dogs a poor model for cardiovascular disease, similarly a poor model for type 2 diabetes. But I actually think this makes them a terrific model because it creates an opportunity for us to really understand, using the kind of approaches that were described in the first 2 talks. Thank you, by the way, for those 2 talks this morning were a beautiful setup. Understanding things like mitophagy, mitochondrial function, as well as systems biology, maybe we can really understand what is protecting dogs from cardiovascular disease, and of course nutrition as well. We're also really interested in frailty, and in human geriatric medicine, we have very simple, carefully thought through, and powerful definitions of frailty. In the veterinary community, you know it when you see it. A dog comes in with declining energy or limited mobility or cognitive changes, and you can say that's a frail dog. But we don't have formal definitions, and we don't have good data, good science, saying what are the consequences of that. So one of the things that we're doing with the Dog Aging Project is to try and develop something that anyone can measure and to say, here's the predictive power. And some general questions. What's the relationship between frailty and aging? Does frailty cause aging, or does aging cause frailty? We want to make this simple. So last summer, we had a veterinary student come into Kate Creevy's lab. Dr. Creevy is now at Texas A&M. And they spent the summer working with clients on really simple measures that anyone could do. Morphological measures to define obesity really carefully, and mobility measures. Here, for example, are some data on the speed with which dogs could climb stairs. And what I've done is plotted them by age quartile. So each dog might have been in a different breed, but we scaled them by the life expectancy of that particular breed. And you can see, not surprisingly, stair climbing speed declines with age. And also, although I haven't shown the regression line here, if you look at weight, removing the effects of age and size statistically, we find a significant negative effect of weight. So larger dogs, not surprisingly, are slower stair climbers, getting back to that question of obesity. So these are sorts of things that we're very excited about working with not only with you, but also with your clients to come up with ways of measuring frailty. And then in the dog aging project, which I'll talk about at the end of the talk, being able to use those data to predict the future for that dog. So ultimately, I'm interested in understanding the genetic and environmental determinants of healthy aging. That's the goal of the dog aging project. We're doing some genetic work. I don't have time to talk to you today about our work on the epigenetic clock in dogs, although that's something I'd be happy to talk with you afterwards about. I'll tell you a little bit about our efforts to understand a little bit about the environment. We haven't yet started working on nutrition. That's a huge challenge, and I welcome suggestions about ways to do that in our population. We have been working on a simpler question, which is how sterilization affects aging. As an evolutionary biologist, this is an idea that I know goes way back. Darwin talked about it in Origin of the Species. He said that nature is forced to economize. If you spend on one side, there's a cost on the other side. You can't have your cake and eat it too. There are always trade-offs in biology. In the fly world, we've known for a long time that sterilization shown by the red curves in males and in females leads to an increase in survival. So this is just a plot of proportion alive as a function of age. Sterilized flies live longer. People have shown this in other species as well. There's even some data from humans. It's problematic data, but thought-provoking nonetheless. Here's one study from Korean eunuchs from a historical population showing that Korean eunuchs live longer on average by 15 to 20 years than the rest of the general male population. Of course, the historical population of Korean eunuchs were living in a different environment, in a more protected environment than the rest of the population. So there are complications. In dogs, the patterns are clear, at least using retrospective data. And this is important that these are retrospective data. We don't know exactly when these dogs were sterilized. For those dogs that were intact when they reached fertility, we don't know if they reproduced, and if so, how much. But the patterns are really strong. Here are the VMDB data for both females shown in red and males in blue. The intact dogs are significantly shorter lived than the neutered dogs. And in the UK, in the Vet Compass data, intact dogs significantly shorter lived than the neutered dogs. So that's clear. This work was done by Jessica Hoffman. She was a graduate student with me. She's now a postdoctoral fellow in Steve Ostad's lab at the University of Alabama, the most productive student that I've ever had in my lab. She wrote many papers both on flies and on dogs. We're continuing to write together. The interesting thing that she did with these data, she found this pattern, and then she thought, well, we can actually look at the spectrum of diseases and how those differ between dogs that are sterilized or not. And so what we're looking at here is the relative risk of sterilization. Lines to the right show an increased risk with sterilization. To the left, a decreased risk. So the bottom and the width of the bar is the overall proportion of dogs that die of that cause. So the bottom line is that sterilized dogs are more likely to die of cancer and autoimmune disease. Intact dogs are more likely to die of trauma and infectious disease. It's not the case that sterilized dogs are dying of cancer because they live longer, and cancer is a disease of aging. Even when we look at young dogs, statistically controlling for age, we see the same effects. So the spectrum of what dogs die of appears to be very different in sterilized versus intact dogs. Of course, these are all retrospective data. It's critically important now that we do prospective longitudinal studies to better understand the underlying causes of this. So the third thing I want to tell you about is an intervention study that we're doing. Can we actually improve healthy aging in dogs? We already heard about attempts to do this through interventions both in the talk on mitochondria and also through diet restriction. In the second talk, people in the lab have successfully improved lifespan and health span through gene knockouts, through diet restriction, through temperature, through interventions with drugs and individual metabolites. This figure here from a science paper in 2010 is hard to read here. The bottom line is that these interventions affect common genetic pathways that are evolutionarily conserved in yeast, worms, flies, and mammals. So it's to be expected that they may well be effective in other vertebrates like dogs and humans. The single most successful intervention so far with a drug has been with a drug called rapamycin. Rapamycin is commonly used in transplant patients. It's an immunomodulator. It's not an immunosuppressant. In fact, work in humans by Novartis has shown that it improves response to vaccine in elderly humans. So it's an immunomodulator. In mice, whether given at 20 months or 9 months, it increases lifespan. It also improves left ventricular heart function when given for just 11 weeks in mice. So with these data in mind, my collaborator, Matt Kaberlein, who's also at the University of Washington and works on rapamycin, these are his data, thought that it would be really potentially powerful to see if we could improve age-related decline in left ventricular function in companion dogs. So I want to stress that these are all studies in companion dogs, not in canine colonies. So the first phase 1 study that we've already carried out and published was done in Seattle. It was a placebo-controlled study in large breed middle-aged dogs. We didn't want to use young, small breed dogs because we would have had to wait forever to get an answer. So we wanted to use dogs that are already showing some decline. It was just a 10-week treatment at low dose with a focus on safety, but we also carried out echocardiogram at 0 and 11 weeks. The most important result is that we saw no severe adverse events, and there was no difference in the adverse events, things like vomiting, between the placebo group and the treatment group. Very small sample size, it was just about 25 dogs, but we did see a modest, statistically significant, but I stress modest improvement in fractional shortening. This is just showing fractional shortening at week 11 versus week 0, and the treated dogs and triangles tend to be above the line. This is just another way of showing it. So a modest improvement. We gave the owners an open-ended questionnaire. Did you notice anything else? No leading questions. That we just asked them if they noticed anything else. There was some suggestion of increased activity levels in the treated dogs relative to the control dogs. Didn't reach statistical significance, but it was certainly interesting. We received a grant from a family foundation to carry out a phase 2 study. This is being carried out at Texas A&M. We don't, as you know, we don't have a vet school in Seattle, so it's difficult to carry out a study at a high level. So Creevy is doing this at Texas A&M. We'll be enrolling 65 dogs over a 6-month period. We just enrolled our first 4 in the last 2 weeks. This will be a 6-month treatment of rapamycin, and at 0, 6, and 12 months will include physical exam, echocardiogram, ECG, blood chemistry, and we'll survey the clients every 2 weeks. And here, the focus is on safety and on heart function, left ventricular heart function, but we'll also be looking at things like the metabolome and the microbiome. So to finish up, I want to tell you a little bit about the Dog Aging Project. And this is really all of what I've told you about so far is kind of laying the foundation for what we hope is going to be kind of a game changer in the study of aging in dogs. We're not the first to think about studying aging using longitudinal studies. The first, actually, studies were in Britain, the canine birth cohorts that have been carried out every decade or so, roughly. Tremendous set of studies. Here in the US, the first study, now the most well-known probably, is the Framingham Heart Study, initially focused on heart function in 1948. Now the grandchildren of those first participants are enrolled. The first aging-focused study was the Baltimore Longitudinal Study, and there are others, of course, the Harvard Nurses Health Study, the Women's Health Initiative, which is Garnet Anderson study from the Fred Hutch in Seattle, and many others. The first sort of 21st century versions of these studies have begun to incorporate the systems biology, or omics, that you heard about in the previous talk. What was the Precision Health Initiative, renamed All of Us, is an NIH-funded study that's going to try and get electronic medical records and genomics from 1 million people in the US. Genomics England has 100,000 people, and then there's a private study from the Institute for Systems Biology in Seattle that's trying to get 100,000 people electronic medical records and omics. The first canine study of this sort is the Morris Animal Foundation's Golden Retriever Lifetime Study. This is a more focused study, 3,000 golden retrievers focused on cancer, completed enrollment in 2015 with dogs from all around the country, a lot in California, Florida, and not surprisingly Colorado, which is where the Morris Animal Foundation is, and also where Rod Page is, who's the scientific leader of this project. And we've been working closely with them to help us develop our study. So part of the rationale of the dog aging project is to really parse apart this equation. The challenge of this equation is that we can identify genes that are associated with complex traits. For example, here we're looking at a plot. Each one of these points represents a genetic polymorphism. There are millions of them in the human genome. And if you look at the relationship between these polymorphisms and lifespan, as is shown here from this 2014 paper, 1, ApoE, is the most highly associated with lifespan. P-value is 10 to the minus 20, but not much variation is explained. Highly statistically significant, but not so biologically significant. This distribution from a 2010 review for these kinds of polymorphisms associated with height, Crohn's disease, breast, prostate, and colon cancer shows that the top hits typically explain between 0.1 and 0.5% of all the variation. So they don't explain much of the variation. Here's the problem. Genotype is up here, phenotype is down here, and the distance between the two is astronomical. So to bridge that gap, this is thinking back to the previous talk, we need to use omics. We need to look at the transcriptome, the epigenome, the proteome, the microbiome and metabolome. These are the critical things because these make up the last steps, the building blocks, especially the metabolome, all the small molecules in the body of the phenotype, of the physical and the and the functional pathways. So this is the goal of the dog aging project, to fill in this black box. And of course, the arrows go in both directions. Just another way of picturing this is that we want to look at genes, measure all the environmental factors we can, measure age, and ask how those affect the endophenotypes, the omics, the metabolome and microbiome and transcriptome to ultimately affect aging. And of course, nutrients are up here in the environment, absolutely critical. So just in the last minute or two, I want to lay out what we're going to do. So the Dog Aging Project currently has a large U19 proposal in review at NIA. Cross your fingers and toes for me. We'll find out in June or July. The grant is to study 10,000 dogs around the country, all breeds, all ages, all degrees of health status. That 10,000 group is our foundation cohort, all of which will be genotyped, and we'll also have other information that I'll tell you about in a minute. We'll have a subset of 1,000 or 1,100 precision cohort dogs, and within that, a subset of about 500 intervention cohort dogs. So all 10,000 dogs will attempt to define aging, measures of frailty, electronic medical records, physical function, demography, reproduction, mortality. For the precision cohort, we'll also include not only the genomics, so we'll have genome sequencing for all 10,000 dogs. For the precision cohort, we'll also look at the epigenome. This tells us about the potential for genes to be turned on or off, and it responds to things like nutrition. So it's a really important readout that looks at the interaction between genetics and environment. The microbiome and the metabolome, which thankfully you've already heard about, so I don't have to tell you anymore. We're excited about involving the clients in this with in-home measures, activity monitors on all the dogs, measures of cognition using a website called dognition.com, which I encourage you to check out. It's from Brian Hare at Duke University. Mobile health using smartphones, measures of physical environment, air quality, water quality, social environment, both human and non-human animals in the environment. 500 of these dogs will participate in a long-term longitudinal rapamycin study. And then finally, I want to tell you a little bit about the outreach opportunities. So I have to say, I think we are in the midst of a crisis in science and society in this country, and it has nothing to do with current politics. It's one that dates back many years. People don't really trust science. We heard about clients going online, finding out information, and going to the vet and saying, here's what I'm going to do with my dog. Not based on evidence, but just based on something that was read online. And we hear about lack of trust in science. So the idea is that because people love dogs, we're going to invite them to be a part of this project in a way that has them doing science without them really realizing it and learning to love that process of doing science. Of course, our first interactors are going to be with the veterinarians. The veterinarians play a critical role in helping us to collect the data. All of the data will be publicly available. We will be collecting hundreds of terabytes or petabytes of data, which we will put out to the world, other than data that are protected by privacy laws. We don't have enough scientists on our team to do all the analysis, so we'll welcome scientists around the world. We'll also be able to provide veterinarians with the data that are relevant to their clients about the dogs at their practice, as well as the opportunity to compare those data with all other dogs in our study. We're also excited about sharing the data not only with other scientists, but in the classroom. We can, for example, create virtual laboratories at community colleges, which often don't have the funds to create physical labs, and we can teach aging, animal health, data science, statistics for no charge, because all you need is a computer and we'll provide all the data and the framework for that. And of course, we're excited about getting these ideas into the classroom and back to the client. And ultimately, as I mentioned, the number one motivator that we think will move this work forward as we start is the fact that people love dogs. So the opportunity to pull in clients and get them excited, have them come to you and say, I want to be a part of this project, or have you as veterinarians be able to say, come to my practice because we are a part of the dog aging project, I think is really a win-win for everybody. And I just want to finish up by thanking my lab, which is many of whom are part of this project. We work on both flies and dogs, but especially this enormous team of scientists. This is not my project, it's our project. Really takes a lot of different expertise, highly multidisciplinary, and a variety of funding sources that have supported this work. And I just have to say, I'm really excited about talking with you. I had a tremendous conversation over lunch today about work that I might be able to do with people here. I'm very excited about it. So I'm looking forward to more conversations with you, and I just want to thank you again for your attention.
much. Hopefully we can just take a couple of questions and then maybe continue the discussions later on at the meet and greet. The microphones are available. A couple of quick questions.
Thank you very much for a great talk. Something I've always been interested in is the fact that pet dogs share the environment with their owners, and I'm not sure whether I picked it up or not, whether or not there's going to be correlation with data that's collected in the same household with different species and how that might pay into everything else that you're talking about.
So we already know some things like the human microbiome is actually an excellent predictor of that individual's dog's microbiome. You can put owner and pet together just by their microbiome. We have, in the US, we need institutional review board approval to ask humans questions, personal questions. We have IRB approval to ask about the health of the owner. We also are going to be partnering with one of the leaders of the Baltimore longitudinal study. Our hope is to enroll to have some of our dogs be part of be in homes that belong to people who are in the Baltimore longitudinal study, so they're in homes where we know so much already about the health of the owner. We're also really excited about the idea of dogs as sentinels. So for example, is it the case that where dogs present with endocrine disorders, that their owners are also more likely to have endocrine disorders, pointing to risk factors in the environment? So that's something that we're very excited about.
Sorry, here. Very nice. I was curious about the data on rapamycin. Are you able, or will you be able to link this with the autophagy marker, autophagy cues in the process, knowing that rapamycin, or are you happy enough to know that is a chemical mimicker of calorie restriction?
So in the first study, we don't have those data. It was primarily focused on just safety. We are collecting blood samples at all time, multiple blood samples at all time points, as well as fecal samples for the phase 2. And I don't know if Matt is planning on looking at autophagy, but that's certainly something that we could do. I'd be really happy to talk with you about it if you'd like to potentially work with us on that.